You want some pretzels?
No, thank you. The checker card.
The card's almost, how much is it going to be? You know?
I'm not sure.
Uh, Dr. Tano, can I borrow you for co-host since Dr. Reno's in Vienna?
Yeah, okay,
Okay, thank you.
sure.
Yeah,
You can eat whatever you have and do the preparation tomorrow.
Yeah, let's mark it up, please.
okay. I have a video. He sent me a video.
Yeah,
Tip of the week, yeah. So you're not going to be deprived of the tip of the week, everybody.
yeah,
Okay, well, we got a few of the main players on here. So Dr. Quinn and Dr. Tano, you're going to get your first email's going to go out tonight about the conference. I know, finally.
yeah,
We finally got it all together. We have the speakers list. We have the hotel, discount. So we got everything on there to go. And it's all ready to go. So you should be getting it. It's going to go out hopefully while we're talking here. Okay.
yeah,
We have one sponsor already too, also. So Sierra Pharmacy in Reno is the compounding pharmacy here.
yeah,
So if you so I'm trying to get a couple of different looks to it. One for the homeopath, so it says homeopathic credits, and then if you guys need a separate look, let me know. The research guys. Otherwise, it's it's got like all our names as the sponsors, and then it's got registration, and that it's all centralized and it's all ready to go. Okay.
yep,
So that's that. If you want, so we've been playing around with it, and so can you see my screen?
no, not yet.
Okay. Hang on. Okay, Dr. Tano, can you take the anybody who to admit folks, and I'll get this up.
Yep,
Okay, so here's the website. It'll be going out tonight. And we have our name. We have Integrative Health Alliance. That's what we're using for the research guys. If you want something else, let me know. Nevada Board of Homeopathic and Medical Examiners, Nevada Board of Integrative Medicine, Homeopathic, and then our other fearless leader is here. Okay, there's a contact page. I'm going to put something in here. I'm not sure what just yet. So here's a registration page. And so we have physicians, virtual, active military, retired, homeopaths, students, lay people. Okay, if there's anything else you can think of, let me know. We got these pictures so far. We want all the speakers to send pictures in with a short bio, and then we'll add them into the speakers list. Here's the schedule so far. Like I said, Thursday is the research folks day. Friday is going to be the homeopaths. We sort of overlapped a little bit. We got Dr. Quinn. I threw myself in there. I wasn't going to, but I did a lecture on 50 years as an anti-aging physician from the Pritikin Clinic to regenerative medicine. So I go through 3 or 4 highlights of things that I've done in each decade. I don't know if that'll be interesting or not. We'll see. So here's the list so far. I'll put this right now is this is the whatchamacallit schedule. This is still the play one. I still have a call for speakers. We could probably squeeze in one or two more at the moment. Sponsors, if you guys have sponsors, please let us know. We got 500, 1,000, and 1,500. I mean, we're not a big group. If for 1,500, we're going to let them sponsor lunch and have them do a non-CME talk. The CME credits, Dr. Alas has given me a little bit of grief because he doesn't quite get the concept of 20 credits can't cost $500. So here's the venue. It's the Element Hotel. Reservations. And there's a little map here. There was a picture before. I don't know what the hell happened to it. There should be a live registration here somewhere. I don't know. Looks like oh yeah, you have to hit this. Okay. It's one of these sort of downscale Marriotts, 179 a night. I mean, last year in Vegas, we used to get 249 at the Palazzo for A4M, and last year it was like 549 they wanted. So this is the hotel. And whatever else is there. So if you want, you got me and Dr. Burgess in there. If you want the pictures of all the prominent folks, in the about, top 2 or 3 in each one of your specialty units, let me know. We'll put those in. So that's what we got. I'll put it in the I'll put the home in the, it looks like it's live now, actually. Okay, I'll put it in the chat, and we'll let you take a look at it, and it's going to go out to our database, the homeopathic database. You guys, if you can, lend us your mailing lists, we can get a fairly nice crowd. It's inexpensive as far as conferences go. I think we got a lot of good speakers, and I know the reason, as you know, is that it's a rush job is because of the homeopaths in Nevada need CME credits by the end of the year. Conference website. Okay, there it is. Okay, it's in the chat. Okay, it is a little bit after 5, so I have a Dr. Nario, who usually does our tip of the week, is in Vienna speaking at a conference, but he sent me a video, so I will share my screen and turn this on, and we'll do that first. Okay, so there he is in Vienna. It looks like it's cold there already.
Hello, everyone. Greetings from Vienna, Austria. Between St. Stephen's Cathedral, the Hofburg, and figuring out the train system here, I've done my fair share of research on Wiener Schnitzel, apple strudel, sacre torte, and real espresso. And I also just enjoyed a night of opera just today. But between the bites and sips, I discovered something new to me. It's called hemopyrolactamol.
Dr. Bia, we don't see him though. Are we supposed to see him?
In wine.
You don't see him?
No.
Let me figure it out here. Okay, in the meantime, I don't know. I have it. It says screen sharing. Stop share. And did you hear him?
No, we heard him before, but his image was not there.
Okay, well, let's see. Got here. I do. You see him now?
Not yet.
No,
It says I'm screen sharing. I'm looking at him and it says I'm screen sharing, so.
Now
I
we see
him
him.
now.
Now it,
Oh,
oh,
he
now it's
now it's
disappeared.
gone.
And now we're not.
We saw him for a brief moment.
Okay. Try it now. You see him now?
Not yet.
Yeah, I see him now.
Yes.
Okay, all right, let me start over.
Yes.
Dr. Changaris, you get any idea of what kind of outfit you're speaking to tonight?
Hello, everyone. Greetings from Vienna, Austria. Between St. Stephen's Cathedral, the Hofburg, and figuring out the train system here, I've done my fair share of research on Wiener Schnitzel, apple strudels, sacred torte, and real espresso. And I also just enjoyed a night of opera just today. But between the bites and sips, I discovered something new to me. It's called hemopyrolactamuria, or HPU. It sounds like a fancy Austrian wine, but instead of leaving you tipsy, it leaves you anxious, fatigued, and zinc deficient. So this is something that I found out here in the conference just recently in Europe or Austria. So HPU is when your body makes too many pyrols, byproducts of hemoglobin. Problem is, these pyrols clings to zinc and vitamin B6 and drags them out of the body. Even if you eat well, you end up with functional deficiency in these specific nutrients, which messes with neurotransmitters, hormones, detox pathways, and stress resilience. Classic signs: anxiety, depression, mood swings, fatigue, PMS, poor dream recall. So remember that one. White spots on the nails and stress incontinence. It often flares at hormone transmissions such as puberty, pregnancy, menopause. So it fits right into our anti-aging world. So why care? Neurotransmitters drop here because it lowers down serotonin, dopamine, and GABA. Oxidative stress rises. Zinc loss leads to accelerated aging. Hormones wobble. PMS, perimenopause, adrenal fatigue, and detox pathways alters. Mold infections, toxins hit harder to our system. And here's the kicker: HPU is now joining our favorite complex diagnoses, the ones where a storm of symptoms appear from nowhere, like Lyme, mold illness, mast cell activation, and chronic fatigue. Even at this early but intriguing, there are studies out there. Warren et al. 2021, the systemic review, showed pyrols higher in psychiatric patients versus controls. The Riordan Clinic actually elevated pyrols linked to zinc B6 deficiency with improvements on replacement. A Mikarova study, higher pyrols in schizophrenia, bipolar, and ADHD cohorts. So not a gold standard study yet, but clinically meaningful. Testing, yes, there is. The urinary cryptopyrrole test. Just remember, samplers are light sensitive, or your patient's pyrols vanish like my strudels I ate yesterday. Treatment is safe and practical. B6 in the form of P5P, 25 to 100 milligrams daily. Zinc picolinate or citrate, 25 to 50 milligrams daily. Magnesium, 200 to 400 milligrams daily. Antioxidants, vitamin C, 500 to 1,000 milligrams, vitamin E, or glutathione precursors such as NAC, plus stress management and anti-inflammatory diet. So HPU isn't in mainstream textbook yet, but in integrative medicine, it's a valuable clinical lens, especially when mood and hormones and fatigue don't add up. And with that, I'll sign off, coming to you from cold Vienna, here in the coffee shop, full of schnitzels, strudels, and good coffee to deliver you your tip or leak of the week. See you again next time.
There you go. So Dr. Nario's giving a okay. So Dr. Nario's giving a talk in Vienna at I think one of the universities there. So tonight's topic. So our speaker tonight is David Changaris, and his topic is muscle microbiome connection, reversing aging with related muscle decline. I got a hold of Dr. Changaris's CV, and wow, what a superstar. I'm not going to read all of it, but it's really quite impressive. The best part, of course, is that he did his internship and some of his fellowships at Penn State University. And so I was there in 2000 to 2002, Dr. Changaris. I worked with the trainers with the football team as an acupuncture assistant of all things. So it was after the decline, and it was during the time when Jerry Sandusky abruptly retired, so nobody knew why. And it was after the heyday of the football game. So, but Dr. Changaris has patents, honors and awards, board certifications, university faculty positions at the University of Texas, University of Louisville, FDA registrations, 1, 2, 3, 4, 5, 6. Did I get 6 or 7 different patents on different types of items? Articles, too numerous to even mention, chapters in books, case reports, abstracts and letters. I mean, it just goes on and on and on. I mean, when did you have time to sleep or eat or anything? I mean, it was unbelievable. So I put a synopsis in the email this week, and it really does not do him justice. So, Dr. Changaris, I'd like you to why don't you just introduce yourself and take it away. You are muted at the moment, though.
Unmuted. Okay, I'm unmuted now.
You are good. You are good to go, sir.
All righty. I don't know how to. Do you have I'll talk for a moment, but do you have the
slideshow?
The slideshow, or do I need to put it up?
Usually, we like the speaker to do it so they can
Okay, I'm prepared to do that.
If need be, I can do it get it up and bring it for you.
No, I should be able to do that.
Whichever,
okay, and I'm having fun getting to know all you guys. It's really a pleasure to meet people who are trying to survive in this wonderful world where medicine is more interested in indications than results. I suspect you folks are more interested in results than indications. It's kind.
I think we kind of like both, but most of our crew have been beaten around by the powers that be in one form or another. In fact, our whole society was canceled by the AOA in 2022. We were summarily drummed out of the AOA after 50 years as a subspecialty.
Yeah, I had a similar, probably more than one similar moment, but the one that around that time similarly, because of the nature of COVID and the work I'd done on the brain renin-angiotensin system, I was able to sort of guess that a molecule that I've been working with would be effective at killing COVID virus. Mm-hmm. And so I tried to present this to the feds, and I didn't know at the time how much of a thumb was on the scale. So I pushed pretty hard and then got nowhere, obviously. Mm-hmm. But I had a solution that would effectively treat COVID, and by nasal either spray or nebulizing, and very simple. How it came about is the family doc was given the terrible job of putting people to bed, as it were, who were turned away at the peak of COVID because the ICUs were all full. And these were all people who were obese, hypertensive, mainly of color, but they were definitely people who were more likely than not to die. Many of them had difficulties with hypoxia. And Sven called me up and said, I hear you're doing something with, did we just go dark? Have I been cut off?
No,
Okay, okay.
you're not good. We're seeing a
Okay.
side view of you though. Is that your best side? We're not seeing a front one.
I can sort of get like this. The whole thing, I ended up sending him the material, and he treated about somewhere between 100 and 200 people, and they all survived. We tried to, he did get it published several places, but when his parent organization where he was working from found out about it, they wouldn't let him publish it, even though it had been accepted. He notified them that it was going to be published, and so they spiked the paper back then. And somehow or another, all the publications that were out in the different places all disappeared for about for many years. It still exists in an obscure place in Europe, but because of all the folderol that's gone on, Sven was finally given permission to do a retrospective on these patients, and so he's in the final stages of publishing this, and so it'll be fun to see this one finally get out. And I'll explain to people the chemistry of it. It is straightforward. It is biochemical, but it is straightforward. And I developed dystonia back in 1997. So, and since I had such a time doing research, I sort of basically chose to do outpatient trauma. No one else bothered to doing this, at least in the medical community. And so I've been treating brain injuries for the last 25 years and noticing that they're all developing stiffness and fulfill the definition of Parkinson's disease. And out of this came a lot of the work that you're going to see today. And I hope it's fun for you. It does invite you into a new world where there are lots and lots of opportunities to be very helpful to your patients. I'd have to say, as a neurosurgeon, my life was pretty bleak. I mean, rarely did I ever see somebody really get better, especially in the kinds of people I treated at the university were moderate to severe brain injuries. So these people never got better. At least they certainly didn't go back to the kind of profession that they did. But the mild traumatic patients that I was able to pick up and document, that you can really help those people. And it's been fun to cobble simple tools that are repeatable and make some sense. So that's where I'm at right now. I'm trying to come out of the closet, if you will, on some of the work that I've been doing, because it unfortunately or fortunately it has more value than my ego, so I have to put it out instead of just let it shuffle into oblivion. So that's what I'm here to do today, to try to explain what I've been doing off in the backwoods countryside of Louisville, Kentucky. Does that help? Hello?
It does.
Okay, good. I'm ready to begin when you are.
We're ready.
Okay. Then I'm going to put this up, I hope. And from beginning, can you all see this? Can you see my slide?
No, see you.
You see me, okay. Let's see what's here going on here. There has to be a way to share the screen. Share here, screen. share. Okay, can you see the plan now, everybody?
You got it.
Okay, from the beginning.
Yep, says the plan.
And we'll make it big so we don't strain our eyes from the beginning. Is this clean for people?
Yep,
Muscles?
looks fine.
Okay. I think the muscle spindle is a neglected part of the anatomy, and it has enormous clinical significance. And I beat my head against when I started treating people with extreme rigidity. When I would reverse rigidity, they basically couldn't walk. They were using their rigid muscles, and so what I had to do is literally slow down the treatment of the reversal of the rigidity that we have in place, so that they could learn how to innervate their muscles again. And that would take months. And I finally sort of played a detective and have come down to believe that this is muscle spindle going on. And it ties back to some of the work that I was exposed to when I was a resident, when I was at Charleston, South Carolina. Way back then, the NIH made the Medical University the southeast catchment for all spinal cord injuries. So we were seeing anywhere from 2 to 6 spinal cord quads or near quads a week. It was a huge service. We had a 9-bed neurosurgical ICU. The general surgeons refused to come into it unless somebody needed a chest tube. And nowadays they have teams of dozens of people doing what a lowly resident was doing, taking care of all these people. So I got up front, very personal with these people who were quadriplegic, and they would invariably develop severe muscle atrophy very quickly over the first 6 weeks. I'm going to talk about these points, the clinical definitions of rigidity, and why I think it points to the muscle spindle. We'll talk about some of the structural anatomy of the tendons and as it relates to rigidity. And then we're going to take a step off into probably a place you haven't walked into, and that is the influence of the skin microbiome on muscle tone. And some of the novel therapeutic approaches that you can have profound results if you incorporate such. And bring up a word that is going to get to be really important, and that's quorum sensing agents. I think this is what's going to happen in the next 10 years. So let's talk about what rigidity is. For me, I go back to a 2003 meeting put on by the PD pods and the NIH. They were trying to figure out just what rigidity means. Everybody was going crazy trying to get a definition. The most remarkable thing about this meeting is they actually came up with a definition that they agreed to. There's a joke about that you can't get 3 doctors to agree to buy $1 for 50 cents. And doctors earn that. And so I'm just saying they came up with the diagnosis, the definition of rigidity is that it simply is the failure of the opposing muscle to relax. So when you, I'll spend a moment developing this, but when you try to move in one direction, the muscles pulling the other directions are supposed to get silent. And in rigidity, that doesn't happen. And you have to sort of keep that in mind. It's a very simple thing that the muscles that are supposed to relax don't relax. Now, this is a typical thing that probably everybody has seen. where somebody comes in complaining of elbow problems. Now, I offer to you that we blur our eyes when people talk about elbows, because the number of people who actually have tenderness in the joint of the elbow it's relatively small compared to the people who have tenderness in the muscles of supination and pronation. And we'll talk about that because that's really important to the wrist. I'm going to use the wrist and forearm muscles as a kind of prototype. But everything I'm saying here can translate to back, can translate to leg, and foot, ankle, whatever you want to work on. But here you can see he's pointing to this old medial ulnar area, and we all know what that means. Now, I want to reduce the scenario to the simplest way of thinking about it, for me anyway. We have a brain, we have a spinal cord, and we have muscle. And a muscle has an attachment at two ends and tendons that sort of hold the muscle. And these sort of reach in like fingers and hold it. And this thought experiment, if you will, shows that if you have a flexor and an extensor muscle attached to the same point on either sides, what you do is you create pretty much what goes on in the human body. And when one contracts, this green pole will move to the right, and when the other contracts, then the green pole will move toward that one. But that's strictly dependent on whether or not the opposite muscle relaxes. Now, I want you to give some thought to what would happen if these muscles have increased tone. What will happen is you're going to increase a downward force. Here I show an arbitrary unit of pressure because all muscles have a certain force. So this is pulling on, once it's stabilized here and the other one stabilized, together they make a vector that goes straight down and produces a measurement, whether half a kilogram of force or half a Newton, depending on how you want to define it. But nonetheless, the more tension you have, the higher this is. Now, I offer to you that as we age, these muscles, in my opinion, have increasing tone in a borderline symmetrical fashion. But what makes it hard for us is that we cannot relax our opposing muscles. And the evidence of that is loss of range of motion. And every time we try to exceed that, we rip muscles and create pain in the muscle. What I'm going to show you is a way to reproducibly relax the muscles so you can increase range of motion. And if you do that, you can literally functionally turn back the clock. Now I'm going to go over some of the simplest stuff of, muscle physiology 101. This is the so-called myotatic reflex, where you have an efferent coming from the muscle spindle that you excite, and it goes through the spinal cord and has a monosynaptic on the arc synapse on the motor, the alpha motor neuron, and causes the muscle to contract. And there's complex communication, crossed extensors, and I'm not going to get into that, but just understand that muscle spindles have a tone. We have 30,000 muscle spindles in our body, and they fire somewhere between 50 and 100 times a second. That's a lot of information that our brain is constantly being bombarded with. But this is the basis by which our brain can figure out where our body is and how we can catch a baseball or a football or whatever it is that we want to do. And here we talk about, now we're talking about the 30,000 spindles. If you take away the voluntary movement of a muscle, like in quadriplegia, these people develop muscle atrophy and very hyperactive tendons. It almost feels like what used to be a muscle is nothing but a fibrous band. And clinically, you really can't overcome these. If you try to, you'll rip things on the more severe patients. And in this situation, you have an efferent coming from the muscle spindle that synapses on the muscle spindle itself, and it tightens up and creates a feedback mechanism where the muscle itself is in contraction. So the denervated muscles become progressively contracted by the muscle spindle in the simplistic point of view. The muscle spindles themselves create this rigidity. And so it's in both legs or both arms. And if they have it in abundance, they can stand on these. And you can get people to literally walk on their rigid muscles in the first few days after complete quadriplegia, using various techniques. But it's all because these muscles are strictly very, very rigid. Now, I offer to you that over time, the body forgets, if you will, how to trigger the muscles with the traditional efferents that are responsible for voluntary control of the muscle. If you look at this reflex compared to the voluntary movement of a muscle, this myospindle reflex, where you excite this and you produce a discharge of the gamma efferent here. This only takes 30 milliseconds. It takes about 200 milliseconds for you to think of something and move your big toe, maybe a little longer. That means this muscle, if it's denervated, can make this trip 6 times while you think about moving it once. It basically teaches when this is going full blast, it basically teaches the spinal cord to ignore the voluntary input. Now, obviously in quadriplegia, there's no voluntary input, but I offer to you, I think this is what happens in aging, is that we lose the capacity to fine motor control our muscles because the gamma efferents of the muscle spindle are firing without ever being inhibited. I would also like to point out that in quadriplegia, the muscles do undergo serious, severe atrophy in about 4 to 6 weeks. And there's nothing on God's green acres that can reverse this. They have tried everything. You can put electricity to the muscle, you can inject it, you can do all sorts of neurotropic medications and the like, but nothing really stops it. And there's something in the nerve from the brain that makes a muscle be a muscle. Whether this is a hormone, whether this is some kind of peptide that communicates something that we haven't quite figured out, but it's very clear that if a muscle doesn't have innervation from the thinking part of our brain that involves movement, our muscle atrophies. And I think this is the basis for the so-called loss of muscle with aging. Again, the reflex monosynaptic arc of the muscle spindle is 30 milliseconds. Again, the voluntary, typically 100, 200. If you have a disordered muscle spindle, if there is a chemical that activates the muscle spindle, if there is something going on in the muscle spindle on its own, you can't stop it. You cannot stop what a muscle spindle does with the voluntary movement. Now I'm going to bring up something that this is probably coals to Newcastle for osteopaths. For most MDs, this is forbidden territory. We don't talk about interneurons. But interneurons are the nerves that live between the sensory nerves that have cell bodies in the dorsal root ganglion and the anterior motor cells of the body. And inside the muscle spindle, you'll see really complicated feedback mechanisms. This is a wonder that it works. And all of this is under the influence of some pretty powerful neurotropic chemicals, and we'll talk about those. So the 1A afferent does something. The 2A afferent does something. All of these things are important, and they can inhibit the ipsilateral muscle or excite it, and do the same on the opposite side. And I would offer to you that what makes a great athlete, a young athlete, a great athlete, is they can train these things almost to do things by themselves without having to think, without the input of the brain. The players, the chemicals, the neurotransmitters, when you look at the muscle spindle firing, the ones that can shut it off, GABA, aminobutyric acid. I'm sure you all think about GABA a lot. Glycine, a strong one. Dopamine, opioid peptides. Now, we're also going to talk about substance P, because this has can do lots of different things. And we'll talk about that in a moment. But if you're going to excite the muscle spindle, glutamate does it, acetylcholine, norepinephrine, serotonin. So we have many, many, many mechanisms to make the muscle spindle fire. The thing about substance P is that it looks like it's really, really important. And let me explain why. This is a paper that was published just a few years ago. I love histochemistry. I cut my teeth on immunocytochemistry from a research perspective. But this is an elegant demonstration of immunocytochemical substance P in a tenocyte. These are cultured tendon cells where they were able to grow them in a petri dish, a little tiny little, actually little cell. And the cell was mobile so that they could pressurize the bottom of the dish. And when they did, they could produce a kind of a stretch of the tendon and then monitor what was going on in that, in these tendon cells. But all this sort of yellowish green stuff, that's substance P. What in the world, is substance P being made by a tendon cell? And this is from probably something that happens in injury, and it probably happens when it's being stressed by stretching. When substance P is around, it can get taken up by neurons. Now, gold nanoparticles, if you put it on the skin, will be found in the spinal cord within 6 hours if you're a rat. Now, this is a pretty profound thing. The ability of nerves to transport large proteins, even viruses, from the periphery back into the spinal cord and from the spinal cord or from the dorsal root ganglion to the skin is well known. That's what herpes zoster is all about. And rabies gets into the brain through the gamma motor neurons, not through the blood. So these nerves have a complicated system for taking up peptides. If substance P gets into the interneuron space, it will produce sustained firing of the interneuron for hours. And if it gets taken up in abundance, it would take a long time. And I'm going to talk a little bit about Clostridia, because Clostridia has a protein in it that when it gets in it, takes literally weeks and months to get rid of. Now, these are some of the findings of the paper we're dealing with the tenocytes and the substance P. And you can see that substance P increases the metabolic activity of the tenocyte. So these are other known enhancers to correlate to. But substance P by itself increases the metabolism of the tendon cells. And it increases proliferation. So there are some really profound metabolic effects that substance P has when a tenocyte is injured, when a tenocyte is stretched in ways that we don't have full understanding. And if it leaks out into the cellular space around the tenocytes, the nerves will take up these peptides and transport them back to the interneurons and produce sustained effects. Now, whether these effects are inhibitory or excitatory depends in large part which neuron that takes it up. This study, and this is where you can, I hope you can capture this. This is the title: substance P is a mechanoresponsive autocrine regulator of human tenocyte function. And it not only does it increase the production of SP, but it also increases the receptor for the substance P. So there's some real difficult moments here going on when you mess with the tendons of a muscle in injury. Now I'd like to introduce the component that I sort of hinted at, that we have a skin microbiome, we have a gut microbiome. We talk a lot about the gut microbiome. Everybody, it's hard to find a woman in the United States that doesn't know they have a gut microbiome, but they don't think about their skin. And one of the things that's really true is that not only does our microbiome on their skin exist, we have a microbiome cloud that extends out 2 or 3 feet. And if you can literally harvest this cloud of particles from our body. And conventional wisdom of people who actually work in this area is that the distribution of cellular elements is unique to the person that the cloud is around. And you have to ask yourself, why do the bacteria and fungi of our bodies make a cloud of around us? The obvious component, and I'm not going to spend a lot of time here, is that they're communicating. They're doing something out in the air. People, when we walk through the forest in spring or by trees during spring, they give off these wonderful seeds and pollen that make us sick. And we just know that that's part of life. And they'll send out a lot of pollen to get one new plant created. Now, the bacteria and fungus and gram-positive bacteria on our skin, they communicate with each other. And in that, the best system known is the quorum sensing system. That each of the, if you're going to talk about just the gram-negative bacteria, they tend to use lactones. And the lactones it can have varying length doodads on them, and they literally look like keys. Like and I'm going to point out to you that one of the near and dear molecules of alternative medicine is ivermectin, and it really is a lactone. Vitamin C is a lactone, and when you start looking at these chemicals as lactones capable of engaging in direct communication with the bacteria in our biome, all of a sudden things get interesting. The gram-negative bacteria like E. coli actually have receptors for adrenaline. So that means some of these bacteria can literally read whether or not we're excited, and they can make chemicals. We know that Pseudomonas can make a compound that can make a mouse literally stare at the wall for 6 hours. Nanogram quantities of this given IP can do this. We know that and that's called PQRS. PQRSA, I think that's the full acronym. Pseudomonas quorum sensing agent. Nothing dramatic. Now, that very same molecule can also turn off the immune system. And so now you have a gram-negative bug that lives on our body, Pseudomonas, that uses a quorum sensing agent to communicate with other bacteria to coordinate group activity. And that's the definition of a quorum sensing agent, is that the quorum sensing agent is secreted in a discrete amount by a given bacteria, specific bacteria. And so that the concentration of the quorum sensing agent becomes a measure of the density of the bacteria present. And when it reaches a quorum, and it's different for each bacteria, it will engage in group invasive behavior. Pseudomonas will, for example, will develop a biofilm when it reaches a certain point. And when it does, it literally takes 5,000 times more antibiotic to kill than if it hasn't reached that density. But until it reaches that concentration, the bacteria remains silent. And there are many silent in terms of the development of a biofilm. There are many examples of other bacteria and fungi where there are group behaviors. And what I've learned is that you can manipulate these with common, simple molecules that are part of our diet. Conjugated linoleic acid can, at very low concentrations, can turn off the communication between certain bacteria like Chromobacter and prevent the proliferation of the purple dye violaceum. I've done this in my lab. I'm not going to show you pictures today, but that would be a whole other talk. But that's very clear that the molecular structures of conjugated with its diene, and I'll go through that in a moment, are such that they will bind lactones, and they will bind peptides, and they will bind other structures as well. Now, the actual lactones themselves can be used as either agonists, antagonists, or competitive inhibitors, just like any other chemical agent. And we'll talk about that. Now, in terms of, is this significant? Does this have any clinical value? Here is a man who presented to me with an elbow that was driving him crazy. He played tennis and golf, but he was having trouble. He had a tournament that weekend and he couldn't golf, and he was really unhappy. And so he came to me, and he had heard that I had had a magic potion, and could I wave my magic wand and make his elbow go away? And so I obliged him. Now, we've been doing this for many, many months. And I'm not going to show you the 4-minute one. We'll have to do that later. But 90% of this swelling in the elbow was gone at 4 minutes, solely from the application of a quorum sensing antagonist. Now, if that doesn't get your attention, nothing else will. We completed the rest of our treatment protocol, and in 30 minutes, this is what he looked like, and his pain was down to 1 or 2. He went out of the office, finished his golf tournament, and he's not been back to see me, and this is 6-7 months ago, or maybe a little longer now. Now, this is a guy that presented to me from a car accident. He was having difficulty thinking, and he had fractured his second metacarpal here and had been to an orthopedist. And this is how the orthopedist treated it, by bracing his 2 fingers here with these little ties. And you can see this is not a happy hand. And he was having trouble sleeping, and he was taking narcotics, and it really wasn't touching it. And so I asked him if he wanted me to fix it, if he wanted me to help him. Now, this was in December of 24, and I had pretty much established a protocol that was about an hour long to treat people, but I'd already been getting indications that some of the effects were happening very quickly. And so, just as a lark, I decided to interrupt the treatment, which involved multiple modalities, at 15 minutes. And this is what the hand looked like. And you can see there's a marked reduction in the swelling. The fingers look nowhere near as bad. This is 17 minutes later. This is the kind of treatment that I'd been doing months prior to this, where people would come in with limitation of range of motion from accidents or sports injuries. And this guy liked to weight lift, and he couldn't do it. And so he showed up and he said, I want to be fixed. And so I put him through the process here. You can sort of see this is almost 60 degrees here. And an hour later, he look at his face, and look at his face here. He can put his hand, his wrist up against the wall, and he was saying he had no pain. This is primarily from the manipulation of the quorum sensing system in the skin. No injections, all transdermal work. Now, admittedly, there was some infrared work in this. I'm not that I think is super important, but this is not the lecture I want to bring up the infrared side of that treatment. Here we have a schematic of a synaptic bouton. This is the other side of the synapse, and these little vesicles get filled with these complicated soup of synaptic vesicles. You can have all sorts of different things in there, enzymes, ATP, and they get released into the synaptic cleft, the cell membrane. When it gets the right signal, will fuse with the synaptic vesicle here, and then release it into the cleft, and then we get ignition. Now, this is an important thing. Exactly what motivates this synaptic vesicle to bind to the membrane here. There's a universe of information out there, but I want to point out to you that one aspect of the universe deals with tetanus. And what tetanus does, it's a product of Clostridia, of course, Clostridium botulinum, and we all know that we get tetanus from a dirty wound. Say we step on a nail, as a kind of, if you will, an example, a thinking experiment. The infection can be relatively small in the foot or in the calf or whatever, and it will eventually become gas gangrene and things like this. But long before it gets there, the Clostridia secretes minuscule amounts of botulinum toxin, and this toxin gets into the bloodstream and taken up by the interneurons specifically, and this is well known. We know the structure of the botulinum toxin. We know the sequencing of it. We know that there's a heavy sequence that binds a neuron, and then it opens up the cell membrane and inserts a smaller protein fragment that blocks the ability of these synaptic vesicles to bind to the end plate, so that the interneurons that are capable of inhibiting a muscle reflex can no longer inhibit. And this is, if the toxin gets into it, it takes a long time until the entire end plate is replaced. And that may take 6 weeks, 2 months for that to happen. And in that period of time, the person develops lockjaw. So here you have a situation where you have a very small infection distally, say in the foot, and 5-6 feet distant, you get lockjaw, or you get opisthotonates, a generalized muscle contraction where a person arches his back and eventually can't breathe because he is or she is so tight. And tetanus still kills 300,000 people a year. I'm just going to, by way of genuflection, just mention that the Golgi tendon organ does play a role in this. It would be nice if we could figure out a way to make it stronger, because these tendon organs are probably going to be more easy to influence. And I would offer to you that some of the work that is done on the physical therapy side of rigidity, where you do things to strengthen the tendon, you can literally do certain kinds of stretches that are static, where if you hold a static position with 50 to 70% of the maximum capacity, and after about 30 to 60 seconds, the muscle spindle just turns off. And this transfers a lot of information and need to the tendon, and can causes the tendon to more aggressively heal. And I think this is going to have, this kind of activity is going to be tied up with this kind of molecular understanding. I mentioned to you about ivermectin. Ivermectin is considered to be a macro lactone, but I'm going to show you a little bit of its structure that is somewhat speculative, but I offer to you that somebody needs to look at it to prove me that it's not what I point out is probably not real. But I bet you it is. And if anybody were willing to make a $2 bet over 15 years, I think somebody's going to prove that the substance of this to be correct. Now, the quorum sensing agent doesn't directly increase the 1A afferent firing when applied to a muscle. The 1A firing is governed by mechanical stretch, fusion motor activity, not by bacterial. The quorum sensing molecules do modulate the mammalian cell signaling and ion channels. So there's a plausible but unexplored link. But this link isn't necessarily tied to the muscle spindles, but everything that I'm seeing, when you do things to silence the muscle spindles, specifically, when you manipulate the chemicals of the muscle spindle, you can produce profound relaxation very quickly. And that's a large bit of my ongoing research in Parkinson's patients, especially those who have the more aggressive kinds of Parkinson's patients. The lactones suppress excitability through GABA, glycine, and chloride channels. They sort of don't do it directly on the muscle spindle, but it's really important to find out the things that can directly inhibit the muscle spindles. Now, this comes from a paper that I published about, see, what is this? Almost October. So that's about 8 months ago. I think it was published in Curis back in February. And you can see this drawing of the forearm, very stylized. We see the supinator here and the pronator here. And this is another pronator on the, and you can imagine this is sort of like a pole, like there. And this is producing a certain tension, and this is producing a certain tension. And when you examine the extended wrist rotation, you take out the influence of the biceps, and the extended wrist rotation is used in the UPDRS, the Unified Parkinson Diagnostic Research System or something like that. I may have that not fully going. But anyway, it's used by the gold-headed neurologist group, a gold-headed cane neurology group, to diagnose Parkinson's disease by having them look at the extended wrist rotation of rotating back and forth. Now, and that picks up rigidity. You lose the range of motion of the wrist in Parkinson's disease and in trauma. And this is where I've been able to see in my patients, especially those who have had a significant brain injury demonstrated by video nystagmography, which is basically the only truly objective measure we have of brain injury clinically, other than just talking to the patient. Now, if you have a brain injury, we know all the epidemiology that's come out in the last 6 to 8 years that these people develop Parkinson's disease and Alzheimer's disease and ALS, basically difficulty with locomotor activity and rigidity, from my perspective. And I offer to you from my clinical experience that rigidity is like number 1, number 2, and number 3 in terms of the aspect of somebody with a brain injury enough where they had anxiety, headaches persisting for a long period, balance difficulties, and tracking difficulties on eye movement. This person will more likely than not, untreated, develop signs and symptoms of rigidity. I had one patient who was a 36-year-old electrician, came to me, and he had already, like within 6 or 8 months, had already begun to have tremors, very, very mild, and he had significant loss of his extended wrist rotation range. And I told him, I said, you're going to have trouble with Parkinson's disease. And he sort of smiled at me and just moved on. And he came into my office 4 years later, and he had florid Parkinson's disease. And I photographed all this and videoed it. And he clearly had the Parkinson's disease, and he was just 4 years from the car accident. And I had him read the chart where I told him that he was going to develop Parkinson's disease within the next 10 years. And because he had disappeared from my clinic when I told him that, I said, well, what was going on here? And he said, I just didn't believe you. But I've gotten so stiff and I can't hold a screwdriver anymore. And he's having trouble earning a living as an electrician. We're now aggressively addressing his Parkinson's disease with significant improvement, but that'll be another lecture. Now, if we just look at the simple measure of the extended wrist rotation as defined by the AMA guides to medical impairment, the arm is held in extension, and the thumb is vertical, and when you rotate and externally, you're pronating, if you will, or supinating, and if you're rotating internally, then you're pronating, and so you should have somewhere between 70 and 90 degrees of range from the vertical axis. And so if you look at the Curis is a great journal. They will allow you to do a case series of up to 5 patients without having to go to an IRB. As you know, IRBs are a pain in the patoot. They're valuable, they're important, but for early work, it's just a lot of work. And I offer to you that this is a wonderful service that Curis does. So I looked at 5 patients who had bona fide brain injuries on VNG, and I published the VNG results in the paper. And I correlated to usually 2 or 3 years down the road, or maybe a little more, to the onset of rigidity in the forearm, where you can say the right external wrist rotation here is 40 in this case 1, and the internal rotation is 60. And you can sort of see that not many of them are even close to 70, 80, 90 degrees. This was a 2-part paper, one in which I developed a little toy for measuring this, and we did studies with training nursing staff and physical therapy staff on how to measure extended wrist rotation so they could get some sophistication on clinical grounds. You can do this clinically a lot quicker than you can with the toy, but it's a good way of learning how to measure this. Now, in these patients, they were treated on the wrist only with potassium CLA, which is known to be a cross-kingdom antimicrobial. It gets both gram-positive, gram-negative bacteria and fungi. And we just applied it, and minutes later, we repeated the treatment. One is we noted that the supinator muscles over here were usually very tense, and there was specific tenderness in the supinators as they attach to the radius. And there's an anatomical reason for that, and it has to do with there's actually a break in the attachments, and so that you can produce some dyssynergy within the supinators, and so you produce a very specific hotspot in the supinator, and it can be very tender. It'll if you hit it just right, the patient will say, "Please stop," and they will say that's. Well, it's not, global, just that level of pain. They would say if it were global, it would be anywhere from a 7 to a 10, 7 to 10 out of 10. Now, if you apply this potassium safflorate to the skin, you within minutes cause the taut bands of the supinator to relax, and the muscles feel like muscles instead of a bunch of ropes, and the range of motion improves quite dramatically. Obviously, this is statistically good, did all the right statistics for each one, and it's wildly positive. But so that's really the first demonstration I'm aware of in the literature. I'd love to be shown that I'm wrong, that shows that if you put something that affects the biome of the skin, like this potassium safflorate, will get a broad spectrum amount that it will inhibit the rigidity of the muscles of the forearm. Now, in defense of the potassium safflorate or CLA, at very low concentrations, these bind quorum sensing agents. And so in some ways, you might be also watching the CLA creating radiosilence. And I offer to you that this kind of understanding, where you have something directly communicating with the muscle to cause local contraction, has a predicate in the infections that we've talked about with Clostridia. Now, here is ivermectin, and what I want to do is point out that this oxygen and the carbon bonds here makes for a lactone, and you have all sorts of different names for 6-membered and 5-membered and 4-membered, and you can anything over about, I think, 10 makes it a macro lactone, and you can see there are all sorts of structures in this molecule here. But I want you to sort of blur your eyes or focus your eyes, whatever it is you want to call it, and look at this molecule here and this molecule here. And these are almost identical. And this is kind of, if you will, like the structure of adenosine diphosphate, where you have this would be the homologous to the adenosine, and each of these molecules here would be like the phosphates. Now, these lactones, this this molecule here, this bond here, readily catabolized by many cellular processes. So this, if this molecule were floating along and came across a receptor for a lactone, this lactone would break off, in theory, and clog it up, because this structure is a little unusual. There aren't a lot of how shall I say, handles on this for the breakup of this particular lactone. So this big molecule would block a receptor. And then you have another one, identical, that if you cut this off, then this molecule can block another one. Or you get two separate blocks if they both fall off, and then this whole molecule can go somewhere too. So ivermectin is like the superstar of potential at screwing up the lactone communication system of bacteria, fungi, and cells. We've already talked about how tetanus toxin can block the release of glycine and GABA, the inhibitory neurons, indirectly. We talked about the protein, specifically the protein that tetanus Clostridia releases is called synaptobrevin. And this snare protein, this synaptobrevin gets inserted into the interneuron, and it blocks the snare protein, which is essential for the synaptic vesicle to fuse and neurotransmitter release. This is a highly specific activity. One neuron, one interneuron is enough to kill you. One synaptobrevin. This is pretty specific. And we only have 10,000 different bacteria and fungi in different parts of our body, for as many as 30 to 50,000 on our skin. And I've already shown you that if you disrupt the skin, you can cause muscle relaxation, you can cause reduced swelling, and you can cause reduced pain. That guy with the second metacarpal fracture went off his narcotic that day and didn't go back on it. And he's got full function of his hand right now. Took a little bit of while, and that's a story by itself, but he made it back. Stimulating 1B afferents in the tendon does not cause contraction. Instead, it causes inhibition of the same muscle. Just that these are the stars. If you want to, I only put this by way of letting you know that these are the superstars of neurophysiology and muscles. If you want to read these things, all this work was done. There's a lot. We could relearn it. And what's fascinating is to look at it from the point of view of quorum sensing potential interruption. Interesting things can happen and do. Now, substance P is what I'm proposing is super important in aging. It is a neural transmitter, if you will. It is the sympathetic nervous system peptide that causes the sweat gland to make sweat. And it is also involved in pain production. The other chemical that's involved in neuropeptides is the calcitonin gene-related peptide. And all of these have lots and lots of places where the CLA can bind, and I'll sort of show that to you here. Neurokinin is in the tachykinin family, can contribute to smooth muscle contraction. So this can directly influence how your bladder empties. And there is possible ways, and I've seen it, where this can reverse some of the bladder or difficulties in peeing as we age. Now, this is bradykinin, substance P. And you can see there are phenyl groups here, a phenyl group here, and here we have this nice little 5-membered with a nitrogen group pyrimidine. This too will bind the CLA. So we got 4 places where this molecule will bind. Let me sort of show you that this is CLA, and the CLA, it is conjugated linoleic acid, and the key word is conjugated. And there is a world of information on conjugated molecules, because the so-called Diels-Alder reaction, which got these 2 people a Nobel Prize almost 100 years ago, is probably the most frequently used pharmacologic synthetic step, because the Diels-Alder reaction takes straight carbons and makes a ring. But it is destructive. But I'm bringing this up is that not because this creates a destructive interaction and changes the molecular configuration of other molecules, but rather this configuration, this conjugation, shares pi bonds. So this is a pi bonded sharing part of this molecule. And this is like a magnet. It's electron rich. And if you have a molecule that's electron poor, it's going to get attracted to this. Now, these molecules can rotate around this single carbon molecule here. So this whole chain here and this chain can really do some fancy wrapping. Now, as I pointed out to you, and I published all this in the patent literature, so I did publish a paper showing that CLA preparations can meet the EPA standards for hospital disinfection, and that's a very rigorous standard. It took us about 6 to 12 months to do it in our lab to get that done right, and it will kill 10 to the 10th in 30 seconds of these various and sundry bacteria in the right settings. At lower concentrations, I give it to my grandchildren for daily washing. I use it to shave, and I use it also to reduce rigidity when I'm working out on a daily basis. It's a wonderful way to reduce rigidity per se. Now, if you look at the molecule, this is a stylized view of this. This is just a wire stick 3D rendering of this. And this is a phenol group, a 6-membered phenol group with shared pi bonds. And you can see that this is on top of 4 molecules. This molecule here and this molecule here, these 2 molecules here underneath it are the conjugation. So the phenyl group will literally share pi bonds in this. This is the exact structure that the Watson-Crick DNA, adenosine, thymine, guanine, cytosine uses to maintain the double helix. So this is a very common physicochemical attraction, and it stabilizes the CLA, and it is important both for messaging and for disrupting messaging. So this molecule is called a dienophile because it loves conjugated dienes, and it seeks this out. And this was the basis of my speculation back in 2019 when the Chinese published the S1 spike protein. 30 to 40% of the amino acids in that spike protein were phenylalanines and tryptophanes. So I knew that that spike protein was going to be a very strong attractor of CLA, and provided the basis for the subsequent studies that we did at the Antiviral Institute in Utah, where they showed that the low concentrations down to 11 millimolar would disrupt the infectivity of the virus. Now, getting back to this is another patient. This is an 80-year-old man who was helping a neighbor repair a second-story roof and second-story window, excuse me. And he was carrying things up and down the stairs all day, and this knee got red and angry. And he came in barely able to navigate the hallway, dragging his left leg into my suite. And we put him up and did our treatment. And an hour later, this is what his leg looked like. It probably looked close to this after 10 minutes, but I didn't know to look for it at that point. This was in October. And so you can put the timing together. You can say this was one of the ones that the effects were so dramatic. I said, what is going on with the swelling? And then I do believe that the most dramatic effects on swelling are caused by quorum sensing competitive antagonists that are generally recognized as safe sugars and that are commonly found in our diet and can be purchased as such. The implications for clinical practice and ongoing research. I mean, you can reduce swelling, you can reduce pain with quorum sensing agents very quickly, to the point where you literally have to sit down with the individual and say, look, the pain is going to go away with this treatment. The swelling is going to go down. If you go back and do the very same thing again, you're going to really cause problems. You need to take some time off and gradually, I take care of a good number of the people in the ballet community here in Louisville, and they literally have to be put in choke chains because they want to go back and climb and jump and do things right away. And if there's evidence of any ligament disruption or torn tendons or anything like this, you still have a disruption. The pain is significantly reduced, and the temptation is to go and pressurize the joint, and you can get into some pretty significant secondary injuries very quickly in that setting. So, but if all you have is just swelling without evidence of serious tendinopathy or ligamentous disruption, you can have people return to near normal functioning and certainly normal range of motion functioning and limited weight bearing in such a way that you can, with proper nutrition and other staged exercises, really facilitate their return to sports, to the ballet floor very quickly, in days as opposed to multiple weeks. There's need for continued research, obviously, to figure out why this is really going on. I need better protocols, smarter people, more. I need people who actually write things down very carefully and reproduces these things, because it's so exciting that when you're doing this, it's hard to stay on task, to develop repetitive procedures, because you're able to produce so many distinct effects where you can reduce swelling, you reduce pain, you reduce difficulties with range of motion. It's just so much fun to be able to offer this. As I was saying, I'm a neurosurgeon. My patients never got better. This is such a wonderful time in my life where my patients are actually getting better. They come in hobbling and they walk out not hobbling, as long as they don't go back to doing multiple stair climbs. So that's where I'm at. I throw it out to questions.
Yeah,
Some of you have heard most of this before, I think.
I haven't.
Okay.
Anybody have any questions? I have nothing in the chat, which is pretty unusual. And Dr. Nario's not here for the online questions. So anybody from overseas, if you have questions, if you can put them in the chat here, that would be a help.
This is Dr. Patel, and I have a question for Dr. Changaris. I just wonder, how did you use the CLA and how did you use the skin microbiome and ivermectin?
No, I actually have not used ivermectin, but I wanted to point out to you that ivermectin is a lactone, and I would like to use it, but I've limited my studies to molecules that are generally recognized as safe for humans and from the FDA. This is an arbitrary decision on my part, just because I'm on the ledge so far out. I'm trying to limit the number of places where people can come at me, and I think ivermectin is going to be a very powerful molecule if applied to the skin in this setting. I don't know that. I've never done it. I have ivermectin, a bottle of ivermectin sitting on my desk. I haven't gotten the courage up to use it. But the other chemicals that are used in foods, sugars, the gluconolactones, these are well known, and I've submitted patents on over these things. They're readily available. You can glucuronolactone. These are sugars that are used in power drinks.
So would you use the CLA liquid and rub on the muscles in rigid muscles in Parkinson's disease? Would that work?
Parkinson's disease, early on in the first few months, it's like a light switch. It does block tremors very quickly. If it's been going on for a couple, 3 years, I personally believe that there's an infection that extends through the lymphatics of the skin to the muscles. Now, the lymphatics of the skin to muscles is a very specific, uncharted territory. There are probably somewhere between 50 and 100 total papers in this anatomic region in the entire medical literature. As best as I can understand it, is that the lymphatics of the skin is more like a sponge, and it was done mainly with immunocytochemistry, and they can sort of generally collect the lymph fluid somehow, pretty much like a sponge might, and then they coalesce to form something loosely like tubules. And as they get near the muscle, they form valves. And when they're in the muscles, they have the valves. And when the muscles contract, then because there are one-way valves, they pump the lymphatics back into the venous system. But from the muscles to the skin, you're talking about a lymph space that I think is infected with something, and I don't know what it is. And you can feel them. The muscles are literally coated with nodules anywhere from a pea size to a grape size, and these are very, very painful. So I think they're probably loaded with substance P. The source of the substance P, I don't know whether it's autologous or infectious, but in those nodules surrounding the muscles is substance P. And as I pointed, or something that causes really major pain, if you try to mechanically disrupt those things, you'll have a very unhappy customer. Anybody who's done myofascial work knows about these nodules, and they'll tell you that these things are very painful and they can't really do much with them because patients cannot tolerate them. Those people who can allow a myofascial therapist to aggressively approach these nodules can have profound and immediate improvement in range of motion and function. I've seen, it's taken me 3-4 years to develop protocols utilizing quorum sensing molecules to block the pain that results from the mechanical disruption of these nodules. And so I can, with the application of nodules and various physical modalities, I can eliminate about 80 to 90% of the pain. It's enough so that most patients will allow me to physically disrupt these nodules and open them up. Once we break through them, we have immediate improvement with respect to rigidity and range of motion. But remember, the substance P has also probably been taken up into the spinal cord and has long-acting changes in the interneurons and the like. And when somebody's had this going on for 5 or 6 years, I'm wondering if somehow or another the spinal cord isn't burned out. But if you get somebody in the first couple, 3 years, you can turn these things off and produce profound, and that can be sustained for decades.
So what quorum molecule were you using in your patients?
Many, but the ones that I use routinely, because they're cheap and they're readily available, are glucuronolactone and gluconolactone. This is very easy to make solutions, very easy to apply by spray. They also have profound effects on pain. Suppliers, they put these in drinks so that people use them for food.
Do you spray that on the skin, or what do you do?
Yeah, I spray it on the skin.
And do you spray it frequently or just one time or every 5 minutes or something like that?
It gets absorbed pretty quickly. And yes, we have a whole, there's a whole treatment protocol, and it involves mechanical and electrical, and we have a complicated dance, but without the quorum sensing molecules, and we would not be able to do it. That allows you to do massage of these nodules that are so painful for people. They call them inflammatory nodules, things like this, but somebody needs to do a really, really good microbial study of these things. I think they're probably fungus, and my candidate is Malassezia. I could spend an hour just talking about Malassezia, but I just wanted you to talk about the muscle spindles and how they affect tone and how you can interrupt it with a number of different ways, including the quorum sensing agents.
Is it possible to get the treatment protocol that you are using?
I haven't codified it enough. I'm still doing early work. I'm not really comfortable to put that out because I'm not trying to hide it. You'll get substantial relief with the glucuronolactone and the gluconolactone if you use that as a spray. And the CLA soaps are easy to make. Are we still on the CME time here?
We are, just a couple questions and we can turn it off. So someone asked, what is a good source of CLA?
Well, that's why I wanted to
Okay.
know if we were off.
All right, hang on. Okay, well, thank you so much, Dr. Chagonis. Unbelievably fascinating. And we'll see everybody next week, same time, same station, 5 p.m. Pacific, 8 p.m. Eastern. And we'll see you soon.